Okay, so welcome to this next video in which we are discussing the palmitoylation of IP3 receptors. Okay, right, so we've now discussed the structure of IP3 receptors, we've discussed S-palmitoylation. Now what we'll discuss is palmitoylation of IP3 receptors. Okay, so the first thing to say that only the palmitoylation of the IP3 receptor type 1 subunit has been characterized at all. We do not know anything about the palmitoylation of the IP3 receptor 2 subunit or the IP3 receptor 3 subunit. Okay, we do not even know if they're palmitoylated at all. All we know is with regards to the IP3 receptor subtype 1. Okay, right, so let me tell you which cysteines we believe are palmitoylated on the polypeptide of the IP3 receptor type 1. Okay, so basically, you remember me telling you the size of these polypeptides that make up the IP3 receptor subunits. They are 2,750 amino acids. So basically, there is a cysteine atom at position 56, i.e. if you count along the amino acids starting at amino acid 1, 2, 3, all the way up to 56, that's a cysteine amino acid, and basically that is a site of palmitoylation. The next one is a cysteine at position 849. Again, this is going to be palmitoylated by an enzyme complex, which we'll see in a moment. Okay, and finally, the final third one is a little bit more putative. We don't know whether this one is palmitoylated as firmly as we know that these two. We suspect it's palmitoylated, but these two we pretty much know they're palmitoylated, whereas this third one is a little bit more putative. Okay, and this is cysteine 2214. Okay, now we suspect that the IP3 receptor subtype 2 and the IP3 receptor subtype 3 are also palmitoylated, but this is an area of ongoing research and it hasn't been characterized yet. The main reason we suspect they're palmitoylated is that they have at least two of these cysteines at equivalent positions. So for instance, the cysteine at position 56 and the cysteine at position 2214 are most definitely present also in the IP3 receptor type 2 subunits and the IP3 receptor type 3 subunits. Okay, so we suspect they're palmitoylated, but it hasn't been fully characterized yet. Right, okay, so now let's talk about the enzyme which is going to stick palmitoyl groups onto these three cysteine residues. Okay, so it's an enzyme complex that consists of two proteins. Right, so firstly, I should say that the process of S-palmitoylation, i.e. adding palmitoyl groups onto cysteine residues within proteins, is carried out by a family of enzymes known as Dhh enzymes. Okay, and there are 23 known Dhc enzymes. Okay, and the specific Dhc enzyme which is going to be involved in the palmitoylation of the IP3 receptor subtype 1 subunit is known as Dhh C6. Okay, so let me now show you the structure of the Dhh C6 enzyme. So firstly, it is a membrane-bound protein, so it's within the membrane. It has alpha helices which straddle the membrane, okay, and it is within the ER membrane. So let's say this is the ER membrane here, okay. Then basically this Dhh C6 enzyme starts with its amino group on the cytoplasmic side. So let's say this is the cytoplasm and this is the lumen of the ER. Okay, so the amino terminus is on the cytoplasmic side of the ER membrane. Then you have a single membrane spanning alpha helix, like so. Then a second membrane spanning alpha helix. Then a very important domain on the cytoplasmic side here, which we'll come back to in a moment. Then a third membrane spanning alpha helix, a fourth membrane spanning alpha helix, another very important domain here, which we'll come back to. and then the carboxylic acid terminus. Okay, so this is the membrane-spanning topology of the Dhh C6 enzyme. So let's now talk about these important domains that we have here. Okay, so this one here, we'll start with this one. This is what is known as the CRD. Okay, and the CRD stands for the cysteine-rich domain. Okay, so it has a lot of cysteine residues within it. And this cysteine-rich domain is extremely important for the catalysis of the palmitoylation, and in fact all of the DHHC enzymes have a cysteine-rich domain, which is on the cytoplasmic side of whichever membrane they are embedded in, and some will be embedded in the ER membrane, some will be embedded in the membrane of the Golgi apparatus, some will be embedded in the plasma membrane. So whichever membrane you're in, it will be on the cytoplasmic side. Okay, now in the cysteine-rich domain, there is a conserved sequence of amino acids. So this is a long portion of the polypeptide, so it has many amino acids in. But in it, a subset of them, in fact a subset containing 4 of them, has the combination DHHC. Okay, and this obviously is why they're all called the DHHC enzymes, because they all have this conserved DHHC within their cysteine-rich domains. Okay, now to understand what this means, you need to know the single-letter amino acid code. So the single-letter amino acid code D stands for aspartic acid. Okay, now I'll show you the R group of aspartic acid. So I won't show you the full amino acid structure, I'll just show you the R groups. I won't show you the core bits. Okay, so here is the R group of aspartic acid. You have a methylene group and then a carboxylic acid group coming off the methylene group. So basically it's a 2-carbon carboxylic acid group coming off the side of the core amino acid structure in aspartic acid. Okay, now these next 2 amino acids, which are both abbreviated to H, this is for histidine. Okay, right, so let me now show you the R group of the amino acid histidine. So basically histidine, you have a methylene group, and then off the methylene group you then have an imidazole ring. Okay, so let me show you an imidazole ring. So basically an imidazole ring is a 5-membered ring in which you have 3 carbons and 2 nitrogens. So we have our 2 nitrogens here, 3 carbons here. Okay, right, and we have a double bond down here and another double bond here. And this is the basis of the name, because this ring is going to be called an imidazole ring. Okay, so basically the double bond between a carbon and a nitrogen, whenever you have a double bond between a carbon and a nitrogen, that's known as an imide bond. Okay, so this is why it's called an imidazole ring. At least that's the justification for the imide bit here. Okay, now just saturate everything up with hydrogens, like so. Okay, and the other logic behind it being an imidazole ring is whenever you have azo, that kind of means pertaining to nitrogen. Okay, so just like thio means pertaining to sulfur, azo means pertaining to nitrogen. Okay, so dazole is quite nice because it implies that you've got 2 nitrogens, which of course you do have in this ring. So that's why this name kind of makes sense. So imidazole ring. So that's the structure of a histidine amino acid R group. Okay, and then the final amino acid here is a cysteine amino acid. We've already seen the structure of a cysteine amino acid, but just to clarify, I'll draw the R group of a cysteine amino acid. So here's the methylene group, and then off the methylene group you have a thiol group. Okay, so that's the R group of a cysteine amino acid. Okay, so all of the DHHC enzymes have a cysteine-rich domain, and within that cysteine-rich domain they have this conserved sequence of DHHC, which is important in the catalyzing of the palmitoylation of cysteine residues on proteins. Okay, now let's turn our attention to this second important domain here. So this is what is known as an Sh3 domain. Okay, and Sh3 stands for Sarc homology free domain. Okay, so Sarc is a protein called steroid receptor coactivator, And basically this domain, this Sh3 domain, is a domain that you find in loads of different proteins. Basically it's a structure that you can have in loads of different proteins, which is identical, or pretty much identical. Okay, now, the importance of this Sh3 domain is that it's going to bind to another one of these Sh3 domains on another protein which is in the ER membrane. Okay, so let me put this other protein here. Oh, actually I'm putting it the wrong side, aren't I? I'm afraid I can't draw it on the side where it's going to bind to this other Sh3 domain, simply because there isn't space over there. But I'll put it here. Okay, so there is another protein in the ER membrane, and this protein is known as selenoprotein K, and it straddles the ER membrane a single time. Okay, so this is called selenoprotein K, and for short, selenoprotein K is often abbreviated to SELK for short, so cell for seleno, and then K for K. Okay, so this is selenoprotein K. Right, and basically selenoprotein K on its cytoplasmic domain has an Sh3 domain, and what's going to happen is this Sh3 domain of selenoprotein K will bind to this Sh3 domain of Dhh C6. Okay, so you'll get this complex of Dhh C6 with selenoprotein K, basically. And this is the functional complex which will then palmitoylate the IP3 receptor type 1 subunit in those 3 slots, basically, on cysteine 56, cysteine 849, and cysteine 2214. Okay, right. So, let's now talk briefly about why selenoprotein K is called selenoprotein. So basically, the selenoproteins are a massive great family of proteins, and the characterizing feature that you have to have in order to be considered a selenoprotein is you have to have a very special amino acid. Okay, and this special amino acid is called selenocysteine. Okay, so let me show you the structure of a cysteine amino acid residue for the 3rd time now, and then I'll show you how to turn it into a selenocysteine. Okay, so here is a normal cysteine amino acid residue. Okay, so again, I'm drawing it. Whenever you say an amino acid residue, you mean the structure when it's in a protein, basically. So I've drawn it as though it's within a protein. This amino group will be bound to the carboxylic acid group of the amino acid in front of it, and this carboxylic acid group will be bound to the amino group of the amino acid behind it. Okay, and the R group of this amino acid is a methylene group, and then with a phyl group off it. Okay, and I'm deliberately leaving a nice space there. So this is the structure of cysteine. To turn it into selenocysteine, you change this atom here from a sulfur atom into a selenium atom, and that's why I left that space there. Okay, so what is the atom selenium? Okay, well, basically, you remember I said that oxygen and sulfur were in the same group of the periodic table. Okay, so if I draw this here, here's oxygen in the periodic table, and this is group 6, by the way. Then below it is sulfur. Well, below sulfur then is selenium. Okay, so here is selenium. And basically, just like sulfur had a similar chemical properties to oxygen, selenium has a similar chemical properties to both of them, basically. So this selenium bound to a hydrogen is similar to a phyl group, which is similar to an alcohol group. So again, it's a similar amino acid, basically. Now, in order to count as a selenoprotein, you have to have at least one of these selenocysteine amino acids within you, and indeed selenoprotein K does. Okay, so basically what happens is in the ER membrane, the Dhh C6 enzyme binds with the selenoprotein K, okay, and they form a functional complex, and this complex will now palmitoylate the cysteines of the IP3 receptor subtype 1, okay, and when you palmitoylate the IP3 receptor subunit type 1, what that does is it stabilizes it in the ER membrane, okay, so let me explain. So basically, the IP3 receptor 1 is a membrane protein. It's it's sitting in the membrane. Okay, now if you attach some palmitoyl groups off this, okay, so let's draw 3 since we put 3 on. Okay, so these 3 represent the 3 palmitoyl groups on a single IP3 receptor 1 subunit. Okay, basically what those palmitoyl groups will do is they'll implant into the lipid bilayer of the ER membrane, and they'll be very stable there. So they will help to hold the IP3 receptor type 1 subunits in the ER membrane, and they'll stabilize them there. Okay, now if you stop palmitoylating the IP3 receptor type 1 subunit, then basically the levels of the IP3 receptor type 1 subunit in the ER membrane plummet down. Okay, and that's because the removal, the speed at which the IP3 receptor subtype 1 subunits are removed from the ER membrane will be vastly increased, basically. So even though you're producing the same number of IP3 receptor type 1s, the rate at which they're being removed from the ER membrane will be vastly increased, and that means that the overall number that you'll have left in the ER membrane will be hugely decreased, basically. And what that leads to is it overall means that there are going to be fewer IP3 receptors. If you've got fewer subunits, then you're going to have fewer tetramers and therefore fewer receptors. And therefore, the response of the cell to IP3, so when inositol 1,4,5-trisphosphate goes up, the response that you'll get to that will be reduced simply because you've got fewer receptors for it. Okay, and this will mean that the amount of calcium that you release in response to the generation of a certain amount of IP3 will be hugely reduced, basically. Okay, so fitting with that, if you knock out this complex, this DHHC6 selenoprotein K complex, either by knocking out DHHC6 or by knocking out selenoprotein K, you stop the palmitoylation of the IP3 receptor type 1 subunit, okay, and that then leads to the expression levels of the IP3 receptor subtype 1 subunit going down in the ER membrane, basically. And we suspect that a similar thing is true for the IP3 receptor subunit type 2 and also the IP3 receptor subunit type 3, but as I say, it hasn't been characterized. Okay, so that now concludes our discussion of the palmitoylation of the IP3 receptors.